Good day, everyone. Welcome to biology class. The objectives of today's lesson are: number one, describe the process of genetic engineering. Number two, explain the process involved in genetic engineering and create a model of rDNA. Today's video lesson is about the process involved in genetic engineering. Let's begin. I will show you some pictures and I want you to identify what can you see in each. Ready? What is it? Yes, these are mice. And these laboratory mice glow in the dark. Next picture. What is it? Yes, these are fishes. Salmon to be specific. And both have the same age. Next picture. What is it? Yes, it's a hen or a chicken with no feathers. Now let's take a look again at the pictures I've shown you. What do you think these things have in common? Yes, the items in the pictures are not in their usual characteristic. A mice doesn't glow in the dark. Bigger salmon as compared to natural salmon. And a live hen with no feathers. But the question is this possible? The answer is yes, it is possible through genetic engineering. But before we discuss that, let us first have a review of our previous lesson about chromosomes, DNA, and gene. What is it? It's a chromosome. A chromosome is composed of long, tightly coiled DNA. What is it? It's a gene. So what is a gene? A gene is a portion of DNA and it's a unit of heredity, which is transferred from a parent to offspring and is held to determine some characteristics of the offspring. How about this last one? Yes, it's a DNA. So what is a DNA? A DNA or deoxyribonucleic acid is a 5-carbon sugar. It is the main component of chromosomes and is the material that transfers genetic information in all life forms. At this point, let us assess what you know about our lesson. So answer the following questions. Ready? What is genetic engineering? A. The process of cutting and replicating of genes. B. The process of changing the DNA of an organism to change its characteristics. C. The process of making healthier food. Or D. None of the above. What part of bacteria can be modified to contain foreign gene? A. Plasmid. B. Cytoplasm. C. Loose DNA. Or D. Cytosol. Last question. This is used to cut a portion of DNA segment. A. Restriction ligase. B. Bacteriophages. C. Restriction endonuclease. Or D. Hormone. Moving on with our topic, there are 4 steps of genetic engineering. We can use the acronym SEER, which stands for S. Selection, I. Isolation, I. Insertion, and R. Reproduction. To better understand the process of genetic engineering, let us make our own rDNA model. How was your rDNA model? You can post a selfie with your rDNA model and send it to this link. Are you aware of the drug insulin used for diabetes? An insulin drug is actually a product of genetic engineering. Of course, genetic engineering is conducted in a sophisticated laboratory with a state-of-the-art equipment. What is genetic engineering, by the way? Genetic engineering is the process by which genes from one organism is transferred into another organism of a completely different species. Let's say a human gene is transferred to a plant, or an animal transferred to another animal, or it could be a gene of an animal transferred to a bacterial cell. Genetic engineering is the process of using recombinant DNA or rDNA technology to alter the genetic makeup of an organism. The core of genetic engineering is the ability to cut DNA and rearrange it in different ways. DNA plus another DNA of another organism equals rDNA or recombinant DNA. Genetic engineering involves the editing of the genomes of a variety of organisms such as bacteria, plants, and animals. These pictures I've shown you before are products of genetic engineering, and they are actually called genetically modified organism or GMO. Genetic engineering is used to produce hormones, vaccines, and other drugs. Genetic engineering has the potential to cure genetic diseases through gene therapy. Another example is a special protein molecule called monoclonal antibodies created through genetic engineering. These are therapeutic agents that attack cancer cells. how is genetic engineering being done? The materials needed for genetic engineering are a specialized cell with a gene that contains the needed trait, a vector, an enzyme that cuts the DNA, another enzyme that will reconnect the strands of DNA, and a target or host cell. A cell containing gene with human insulin is one example of a specialized cell. A vector is a piece of DNA that serves as a carrier for the foreign DNA. One common example of vector is plasmid. A plasmid is a small accessory ring of DNA found in bacterial cells. DNA ligase is used to seal the cut portion. It facilitates the sealing of fragments created by the restriction enzyme. A host cell, or target cell, serves as the site where recombinant DNA is placed. The procedures are the selection of desired trait, the isolation of DNA fragment of desired trait from a donor organism, the insertion of the isolated DNA fragment into a vector, and the replication of recombinant DNA in a host. Let us look on how the insulin drugs are being produced using genetic engineering. So the first step is selection. Select the trait you want to produce. In this example, a gene containing insulin was selected from beta cell. A beta cell is an insulin-producing cell found in the islets of Langerhans of pancreas. Beta cell acts as a specialized cell. The second step is isolation. Isolate the genes from specialized cell. The gene carrying insulin was isolated from the beta cell. Isolation of the gene is done through the help of an enzyme that cuts the gene. Third step is insertion. Insert the gene to the target cell. Bacterial cell, which has plasmid, acts as target cell. Plasmid is temporarily removed from the cell. An enzyme restriction endonuclease is used to cut the plasmid. Any two fragments produced by the same restriction endonuclease can be joined together with the assistance of the enzyme DNA ligase. We have now the recombinant DNA, or rDNA. The plasmid is returned in the bacterial cell. The fourth step is replication. Replicate the new organism. After returning the plasmid in the bacterial cell will undergo DNA replication and the cell will reproduce. Many cells now have the insulin gene and will undergo different processes to produce insulin drugs. Generalization. Genetic engineering is the process by which genes from one organism is transferred into another organism of an entirely different species. Genetic engineering involves the following steps: selection of the characteristic or trait needed, isolation of the genes containing the trait from specialized cell, insertion of the genes into target or host cell, and replication of the new organism. The materials needed for genetic engineering are a specialized cell with a gene that contains the needed trait, a vector, an enzyme that cuts the DNA, an enzyme that reconnects strands of DNA, and a host cell. Production of insulin drugs to cure diabetes is one of the many products of genetic engineering. A genetically modified organism, or GMO, is any organism whose genetic material has been altered and use genetic engineering techniques. Did you understand our lesson? Very good. Now, for application, answer this question. If you are a scientist, what GMO will you produce and why? Send your answer to this link. To assess how much you have learned about this topic, answer the following questions. So, instructions: complete the table by adding word or words in the category and characteristic row. An example is done for you. For part 1, complete the table by adding word or words in the category and characteristic row. An example is done for you. So question, what is genetic engineering? Genetic engineering is, for category, a branch of applied biology that, characteristic, involves the manipulation of gene. So, genetic engineering is a branch of applied biology that involves the manipulation of gene. So, complete the table. For part 2, complete the following sentences. Genetic engineering is, the materials needed are, the procedures of genetic engineering are, I believe genetic engineering is important because send your answer to this link. Let's go back to the pretest. What is genetic engineering? Is it A, the process of cutting and replicating of genes? B, the process of changing the DNA of an organism to change its characteristics? C, the process of making healthier food? Or D, none of the above? The correct answer is letter B, the process of changing the DNA of an organism to change its characteristics. Number 2. What part of bacteria can be modified to contain foreign gene? A. Plasmid, B. Cytoplasm, C. Loose DNA, or D. Cytosol? The correct answer is letter A, plasmid. Third question. This is used to cut a portion of DNA segment. A. Restriction ligase, B. Bacteriophages, C. Restriction endonuclease, or D. Hormone? The correct answer is letter C, restriction endonuclease. As continuation of our topic, for your assignment, answer the following questions. Number 1. What are the advantages and disadvantages of GMO? Number 2. What disease, if there's any, could occur from genetic engineering? Send your answer to this link. And this ends our lesson. Thank you and good day.